I'm having a little trouble hearing as well, but you guys can hear me okay, and you can see my slides?
Yes.
Yes.
Okay, excellent. And Laura might have told you that I have a little bit of a scheduling conflict. So for some reason, daylight savings time changes like 3 weeks earlier in the United States than it does in Europe. So this talk is now scheduled in the middle of my clinic. So I'm sorry I can't stay too long for the rest of the meeting, but I do appreciate the opportunity to share a little bit about my research. So I don't know how much of an introduction you got, but I'm Paula Amato. I'm an OBGYN reproductive endocrinologist in the United States. And I work on assisted reproductive technologies, essentially for infertility, mostly in ovarian aging. And I just wanted to share a little bit about one of our projects this morning about in vitro gametogenesis, which you may have heard about. The fancy term means trying to make eggs basically from skin cells. So I work at Oregon Health and Science University. Like most research teams, have a group of collaborators. My main collaborators, Shootcraft and Home Club, you might have heard that name as well. And as I said, we're mostly interested in treatments for infertility and ovarian aging, but we're also interested in regenerative medicine because we work on embryonic stem cells and in the past several years, some germline gene therapy as well. But today I want to focus on IBG and want to acknowledge our funders for the human work is from Open Philanthropy. As you may know, the NIH doesn't fund any human embryo research, so any human embryo research we do is privately funded, and for this project we were grateful to get a grant from Open Philanthropy. So I'm going to talk a little bit about IBG, what possible uses are for it in reproductive medicine, also how it might change the practice of reproductive medicine if it actually comes to fruition, and that's a big if, as you'll hear, and then just touch on some of the regulatory and ethical issues as well. So who would benefit from IBG? The biggest group, of course, are women who are older. As you all know, women are born with a finite number of eggs. As they get older, the egg number declines, eventually run out of eggs and go through menopause. On average, that happens at age 50, but fertility, of course, declines way before that. In addition, there are women who are born without eggs or for genetic reasons undergo premature ovarian failure, or cancer patients, for example, sometimes who undergo chemotherapy, that can cause premature ovarian insufficiency as well. This can happen in men as well. We call that azoospermia. I'm not going to talk too much about that. Turns out trying to make sperm from skin cells is even harder than trying to make eggs. So that is not something I personally work on, but there are other labs that are working on that as well. The other kind of group that would benefit from this technology are same-sex couples. So at the moment, if a female same-sex couple wants to get pregnant, they would use like a sperm donor, and if a male same-sex couple wants to have a child together, they would typically use an egg donor and a gestational carrier to carry the pregnancy. But this technology might allow them to have a child that's genetically related to both partners. If we could make an egg from skin cell of one of the male partners, fertilize it with the sperm from the other male partner, that resulting child would then be genetically related to both partners. Some other kind of far-out things there: you could reproduce with yourself if you wanted to do that. I'm not sure why. Multiplex parenting, which is the concept where you could have like 4 people who wanted to have a child, and then you could make 2 embryos from 2 of the pairs, and then you could make, an egg from one of the embryos and sperm from the other embryos, and make another embryo. And of course, that resulting embryo child would have 25% genetic contribution from, the 4 people involved. So, just to, give you an idea of the possibilities if this were to come to fruition. This type of graph is probably also familiar to a lot of people as well. We know that the average age of first birth is increasing in the United States and throughout the world. Really, more women in their early 40s are having children, as you can see from this graph. That's largely due to the availability of IVF and assisted reproductive technologies. At the same time, we've seen a decline in teen pregnancies, which is obviously also a good thing. Infertility is super common, believed to affect about 1 in 6 couples. This is latest data from WHO, and that's worldwide prevalence. These are the different reasons. This is from US data on IVF and ART. Different reasons someone might need to use IVF. And you can see here, almost a third of the cases are done for what we call decreased ovarian reserve or decreased egg number. So potentially, a lot of people could benefit from this technology. This is also probably a very familiar graph. If you plot pregnancy success rates in IVF, live birth rates, rather, against age, you can see for patients using their own eggs, you see quite a dramatic decrease in live birth rates as women get older. If they're using donor eggs, however, the pregnancy rates stay relatively high. And that's because the age of the uterus doesn't seem to matter so much. It's all about the age of the eggs. So IVG would allow women to have genetically related children, theoretically, at any age. Now, of course, they can, at the moment, have children at older ages if they're willing to use donor eggs. And we have to remember that even if this technology works and this is a possibility in the future, we do have to keep in mind that carrying a pregnancy when you're older does carry additional risks, right? Basically, any complication of pregnancy, the risk is higher as women are older. So we would definitely have to deal with this issue. Now, one of the potential solutions, of course, you could use a gestational surrogate or gestational carrier, but that has its own sort of ethical issues, and it's expensive, etc. So I mentioned same-sex couples already, and again, this technology would allow 2 men primarily to have a genetically related child, genetically related to both partners. I mentioned it would change the way we do IVF as well. So right now, as you know, women have to take these injectable fertility medications for a couple of weeks and then undergo an oocyte retrieval or egg pickup, different terms used across the world. Which is a minor surgical procedure, but nonetheless, has risks associated with potential bleeding, infection. Ovarian hyperstimulation syndrome is, a rare but serious complication. And then the gametes are, put together in the laboratory, and then the embryo is transferred back into the uterus through the cervix. But if we could make eggs from skin cells, basically this whole process would be happening in the petri dish, and the woman would not need to take these hormones, and would not have to undergo egg retrieval. We just do like a little skin biopsy and be able to grow eggs from that. These are some of the side effects associated with IVF, the way we do it currently, that would mitigate all those things. So in vitro gametogenesis has been done in mice already. A Japanese group published a few years ago. They were basically able to make a blood-borne mouse using this technology called iPS cells, or induced pluripotent stem cells, which is a different technology that we're using, but just to show you that it is theoretically possible in mice. Now, they of course are working to try to do this, replicate this in humans, but it is more challenging in humans, and there's a couple of startup companies as well, most notably one called Conception in the Bay Area that's also working on similar technology to try and do the same thing, to make eggs from stem cells. We're using kind of a different strategy, and it's the we're kind of a place in the field where it's worth trying different things to see what works. So maybe iPS cells will be the way to go, but this is kind of a different strategy which we think has some advantages. So we use basically somatic cell nuclear transfer, which is the same technology that you use for cloning. And we published a paper several years ago. Cloning has been around for a long time. Dolly the sheep, several species have been cloned. Not humans, of course, but we were able to create embryonic stem cells from human oocytes and published that a few years ago. So our strategy is, what if you take the nucleus from a skin cell or any somatic cell, put it in an oocyte or egg that's had its nucleus removed, and then try and get that oocyte to undergo meiosis so that it kicks out half the chromosomes, because a somatic cell, obviously, diploid has 2 sets of chromosomes. If we undergo meiosis and you get rid of half the chromosome, could you then fertilize this new egg now with sperm and, create a baby? This is kind of in graphic form, to remind you what happens during meiosis. So we would take a somatic cell nucleus, try to get it to undergo meiosis, kick out half the chromosomes, then fertilize it, and try and create a baby that way. Okay, that's the idea. So we're able to do this in mice. This just shows kind of the spindle in a mouse oocyte. The spindle is the chromosomes lining up before they divide. We published a paper a couple of years ago and actually got some monoclonal mice. These are embryos down here. Process is very inefficient, but in some cases we were able to get live-born mice. So this shows that it is potentially possible. These are the mice. Again, very kind of inefficient. I'm not going to go through all the data in the interest of time, but I'm happy you can review the paper. This is published, but I'm happy to, if you have questions afterwards, feel free to reach out. Our most recent paper, just published last week, or a couple weeks ago now, we looked at these mice more closely, trying to get an idea of how the chromosomes actually segregate. And we used 2 different strands of mice, so we can kind of map them, if you will. One of the scientists in the lab developed this very clever sequencing systems where we can see which chromosomes from which mice segregated where, okay? And what we found that in most cases, the segregation was, they did segregate, but it was kind of random. So what you want, if you have 46 chromosomes, you want like 1 chromosome going to go there, 1 chromosome going to go there, 2, 2, 3, 3, etc. 20 chromosomes in mice. But we didn't see that. Most of the time, yes, we were getting 10 chromosomes in one egg and then 10 in the polar body, but it was kind of random which chromosomes. So obviously that's not going to be compatible with life. The only time we saw complete segregation was when we were using inbred mice. So obviously a lot of challenges that we have to overcome still before this becomes viable. Just some preliminary data, we're now doing this in humans. So in Oregon, we're very lucky. We can actually pay donors to donate eggs for research, and we can actually create embryos for research purposes. And very few places in the world where you can do that. So we did exactly the same thing. We got women to donate eggs. We got skin biopsies from another woman, took the nucleus out of one egg, this is the spindle here, and put the somatic nucleus in, and then looked to see what happens. And they did undergo some form of meiosis. We could see a polar body. But these embryos, at least so far, haven't really, the frequency of development is really low. And we think this is because of the reason I mentioned earlier, probably getting random segregation of embryos, of chromosomes, so they're not equal percent, and the embryos are arresting very early on. But we certainly can get them to segregate to some degree. We can actually fertilize them, and we can actually watch these embryos develop to a certain point. So at the moment, the next step for this is to try to figure out how to get these chromosomes to pair like they would in nature, and then divide as they would in nature. So you get, 23 chromosomes in the polar body, 23 remaining, but the right 23. So that's where we are. We did similar kind of studies to the mouse studies to see how the chromosomes were segregating. And as I said, they were segregating, but not very normally, which obviously is not going to work. So we can do this in mice, we can do it in humans. It would provide a strategy for treatment of infertility and ovarian insufficiency and allow same-sex couples to have a genetically related child. Number of ethical issues that I'm not going to have time to go into, but obviously, first and foremost, is the child going to be healthy, right? And there's all kinds of challenges like imprinting, recombination, all those kinds of things that we need to study. But then there's all these other social implications that are also very important to be discussing, including who will have access to this technology, which is, the same issue that comes up for all these reproductive technologies. So I don't want to minimize these, but I'm just not going to have time to talk about them in detail, but to let you know that we are paying attention to and having conversations about these things as well. If you're into prediction markets, people always ask, well, how long, before we can make an egg from a skin cell and make a baby? About 10 to 20 years. This is from Metaculus. They're predicting the first baby to be born somewhere around, 20 years or so from now. So we'll see. Some of this stuff may sound pretty out there, but of course, that was true of IVF when it first came online, 45 years ago. And, sometimes you have to do things that are kind of really radical. And then, several decades later, we'll look back and think, oh, can you believe we did IVF this way? We did, not like we're doing today. So, anyway, this is the team. Metalopov Lab, current members, my colleagues in the REI division at OHSU. Here's my email if you want to reach out with any questions or comments. You can find me on X or Twitter as well. I know that was super brief, but hopefully it gives you a taste of what we're up to. And I'm happy to entertain questions, but unfortunately not right now because I have to go back and see patients, but maybe Laura, you can relay them to me later if there are any.